Okay, thank you, Guillermo. And I'd like, of course, to thank the organizers, Siddhartha, Sergio, and Guillermo, for the very nice invitation and for putting this together. I should say my talk slash class, it's quite basics compared to the other ones I have seen. And maybe this one would be better if you have put it at the very first day of the course. But I mean, because I was traveling and all these things that happens to you, okay, only now we are going to have some really basic stuff. Uh, for those that are not too much into neuroscience, then you can get back to your thoughts. Oh, so this is what that guy was talking about. But anyway, so pretty much I divided things in 3 parts. I'm not as sure that we're going to have time to see all these. I mean, I know that I have 2 hours, which is a lot. I mean, everyone is going to be tired. I'm going to be tired. You guys are going to be tired as well. But let's see how far we can get. So for the first part, which I would say can take up to 1 hour, I'll make a very general introduction about what people do employing computational tools for analyzing data, and then I'm going to go and give you some specific examples of classical results, say, that people found when working with the units, the neuronal units. I should say the examples I picked here, I mean, I like them a lot, but they're not like an ultimate word about what's going on, right? I mean, there are plenty of nice results out there that I didn't collect. And same thing for the local field potential. I'm going to show you some examples of what people have been doing. Since I'm here and I also do some simulations, I'm just going to, I mean, for those that are not used to modeling neurons and neural networks, this is just going to be a very quick primer about how people can use computers to gain, to do the modeling. Okay, and so this is going to be the nice part in which I'm going to show you movies and things like that, and going to find that neuroscience is fascinating. And then I'm going to go to my own work, which is duller than these guys here. And then, okay, I'm going to show you. So basically, what brings these two together, so these would be tools that we developed and applied for the data. So it's one tool for analyzing the units. This is work together with Vitor, which is that Brazilian guy that looks like an Argentinian over there. And then, if time allows, I'm going to go to this other measure, which is about seeing how brain waves can interact. This I started during my postdoc years, and I keep doing these days. Okay, so basically, and I'm going to be putting these bold things every once in a while, so we know where we are. But don't worry, I won't pass the 2 hours threshold. We can stop anytime. So the way I see how math can help in neuroscience, I put math here because I did some math training. I have a master's in math. And there are several ways people look at how this exact science can help neuroscience. The way I see it, so this is a personal view, I see that if you know, I mean, by math, hello? Math, I mean, in the very broad sense of math, I mean, even physics tools or computational tools, pretty much there is a lot of programming. The way I see is that we can use math both to help in research, in neuroscience research, from the experimental side as well as from the computational, the more theoretical side. So I'm particularly interested in the so-called brain waves. I'm going to soon let you know what I mean by brain waves. But the thing is, suppose you design an experiment, you record the neurons or record the LFP activity from a given region of the brain, you put your animals to do whatever paradigm you are interested in, and then you get all this time series with the electroactivity of these brain regions you recorded, right? We cannot get this pages long. I mean, it can be a very, very long time series and send to the editor and say, you just published, I did this experiment, so here are the results. I mean, we have to make a meaning out of all this time series. So what people usually do, and this, I mean, there is a lot of work to collect data. I mean, the experimentalists, they do a wonderful job. But after you have your data, you still have to try to find some set of rules, try to find something consistent that can explain what's going on there. And for that, I usually use MATLAB, or people use Python, or you can go straight to C++. But then we use a lot of math. I mean, we program math tools here to try to understand what's going on. So the particular research line I'm interested in, oftentimes we find that there is a pattern of organized electroactivity, meaning that these electrophysiological recordings I have, very often they show oscillations, okay? So this is an experimental research. This is empirical. I put the animal to whatever, take a decision, and I found that during decision, this particular brain wave shows up. And then what people can do is, okay, so we have this finding, and now we can play around with this kind of reverse engineering. Because we know that in this population level, in which the electrode captures, I found, say, my brainwave, and now I want to know what happened in the network or even cellular level that gave rise to this mesoscopic effect. And then you do all the modeling, or you arrange your neurons, your network, you play around with different subtypes of interneurons, and eventually you're going to be able, we have a lot of degrees of freedom when we do modern neuroscience these days, precisely because we don't know much, right? So there are tons of parameters that we are free to choose because we don't have confident measures about them. So I would say, unless you're trying something very crazy, oftentimes you are able to make a network that's going to give you the experimental result you found. But of course, a model is only good when it's able not only to match the data, which I would say is not that hard to match the data, but it has to generate some predictions in which you go back to your bench and try to see whether your modeling prediction worked or not. This virtuous cycle here, this happened very well for physics. Everyone knows that in the last century, physics profit a lot from this. And for biology, I would say in the last 20 years or so, we are starting to build this kind of framework. And besides that, the way I also see math or programming / physics helping is this particular topic here. So this would be a side research line which I have. But basically, it gives you a lot of work to collect this data, right? You can spend 2, 3, 4, 5 years collecting the data. And okay, you can apply some standard tools you have out there, you publish your paper, and but never, of course, trash your data, because you never know when someone goes reading physics and math literature and come out with new tools. So for instance, the work I did during my postdoc, all the data sets I had access to, they were already used. In the sense they already published 1, 2, 3 papers out of them. But I'm going to show in the other parts of this lecture that we came out with new measures and we could re-look at the data and find still new stuff. So pretty much, okay, what I expect from this, I'm from the romantic side. I want eventually to know how does the brain work. Of course, when we write grants, we have to say that we want to cure Alzheimer and Parkinson's and all that. But the way I do it is we can only fix what we know, how it's broken, right? So we first need to know how the brain works naturally, and then we see if we can fix it. This is my symbol for things that are very crazy. There is a lot of theory, there is a lot of mysticism, so pretty much I think we are in a very good period to be in neuroscience precisely because of that, right? That we have much more theory than is backed by data. So we have a lot of big guys claiming that their theories to be true, but to be honest, we have very few solid results in neuroscience. But that's why I think it's good to be working in neuroscience, because there is a lot yet to be done. We don't have big actions. Biology in general, right? We have Darwin law and, well, anyway. So the talk's pretty much about this, what we can do, like analyzing the data, very basic stuff. And in the other 2 parts, I'm going to show you like one tool for analyzing neurons, another tool for analyzing the EEG or the LFP. And I'm going to show you some examples of applications. Okay, so this is just very basic stuff. Then computational analysis of electrophysiological signals. So this is high school knowledge. I know you know that neurons produce electroactivity. So we have the action potentials, we have the postsynaptic potentials. And pretty much when you put an electrode inside a given brain region, you're going to capture as well, even recorded from the extracellular side, you're going to capture these variations in electroactivity. Right? This is okay. If you guys don't know what this is, you shouldn't be. Yeah, anyway. So this is just to illustrate the kind of data I work with. So this is collected from, again, it's extracellular, it's inside the brain, but not inside the cell. So this is called extracellular recordings. And in freely moving animals, right? So the animals, they are moving around while we are collecting these electrophysiological signals. And there are several variations of these, even to this Carmen Miranda-like hats to something. We are trying to get smaller, but it's still difficult. We are trying to get wireless as well, but still. And there are several types of electrodes. This one, Rodrigo mentioned, this is a tetrode in which we have 4 wires glued together. And the idea here is that if you use these 4 wires, every neuron that, emits a spike here is going to have a different signature in each one of these 4 wires, right? Because if you only have a single wire, it could be by whatever. It could have 2 neurons that could give rise to the same spike waveform in a given wire. But when you triangulate, you can play around with 4 wires, and then you can separate better your units. I'm going to get back to this spike cluster. This has to do, of course, with the class by Rodrigo Quiroga. Just to mention as well, inside the cell, we all know high school stuff that the action potential have a very patterned shape, right? It goes down, up and down. But outside, in the extracellular mean environment, the shape of the action potential may vary depending on well, several factors, right? But it's not that standard action potential shape that we get from inside the cell. This is just to promote this electrophysiology. So why do I like electrophysiology? I mean, not only me, a lot of people like. Because I mean, we know the brain is extremely fast, extremely dynamics. I mean, it's always changing, and I mean, our tools the more tools we have to fight these problems and to try to solve them, the better, right? But I mean, I think it's kind of intuitive that once you kill the animal, you get your brain, you put some slices and you whatever, do some immunohistochemistry, the brain stopped working. So decision making and several cognitive processes, they occur in a very fast scale. And the good thing about electrophysiology is that we do have this very nice time precision. Right? Well, I'm no geologist, but this one is just to illustrate that there are several ways in which we can we can analyze our signals. So I remember this cartoon in high school. Here would be the signal you collect, and the analogy here is just to say there is not a single pipeline, right? You can analyze your data in several different ways, and depending on how you analyze your data, you're going to have different products. Then you can analyze further, playing around with statistics and things like that. So actually, I think I just lied because there are two main pipelines, right? There are two main pipelines. So you stick your electrode there, you collect your data. This is called the raw signal. And the two main pipelines that we have in this field of analyzing electrophysiological data is looking at the units or looking at the EEG. So if you high-pass filter your signal, which is represented here, you're going to see that. So this is what we call noise, but it's actually not only noise. But depending if you have, say, a neuron nearby your electrode, whenever this neuron spikes an action potential, it leaves a sort of a scar. I mean, you can see it there in the extracellular recording if you high-pass filter. And in doing so, I mean, if the threshold, you play around with clustering. I'm going to talk a little bit more about this. But in the end, you get access. So we call, by whatever reason, maybe because of Ramon Cajal, we call the neurons, in this field, we call them as units, right? So you have access to, units are neurons, it's the same thing, right? You have access to the units. And pretty much you can start working with the spike time. So you know the times of the spikes of the neurons you got. So this is one of the pipelines. You can spend your whole life here. Say I'm gonna work with prefrontal cortex neurons of monkeys during a working memory task. Some people don't even look to the other part of the spectrum, but some others do. So if you low pass filter your signal, then you have access to this, which is believed to be a sum of a lot of sub-threshold potentials. I mean, actually, this is debatable. There's a lot of things going on here. But people think that these tend to represent a lot of synaptic potentials. But, I mean, the story is more complicated than that. Whether spikes also leak to this band is still debatable. But the point being, if you low-pass filter, then you have access to this. You start working with this so-called local field potential. Local field potential is the same thing as an EEG, if you want to think like that, but it's just that it's collected inside the brain, okay? So this is the local field potential, and what people do further is to band pass these local field potentials, okay, into several different bands, and in doing so, then you start working with the so-called brain rhythms, or brain waves, or brain oscillations. They have these different Greek letters here, so each name is only a frequency band, okay? So delta, theta, alpha, beta, gamma, and many others. The different Greek letters they just denote different frequency bands. You can also play, so here as well, right? There's a lot of people, I mean, I have papers, I only dealt with the local field potential, didn't even look at the units, especially because I didn't have units. But again, you can say, I want to study alpha oscillations, the occipital lobe of meditators. I mean, you can play with this a lot, not even caring about this, but you can also do this kind of combined analysis, right? You can see how the neurons, how they relate to the different brain waves as well. And this is something I'm gonna, if time allows, I'm gonna talk about this. I mean, nowadays I have been talking about how waves of different frequencies, they can interact with each other. Okay, this is called cross-frequency coupling. If time allows, I'm gonna show you some results I got at looking at this. Okay, so this was the general introduction. Now I'm gonna talk about analyzing neurons. So we are here. Okay, so this is the pipeline of the data analysis. So this is a slide. So this is from Quiroga work. So basically, put your electrode there, have a reference, and here you already high-pass filtered, and then you have a very nice, of course, we always put cartoons with this very nice signal-to-noise ratio. And then you can threshold these guys here. So you localize this point, you create a window around one of these spikes, and here you collect several of these spikes. And here you can see that the spikes, they have different wave shapes. Again, inside the neuron, the action potential is pretty much standardized, right? It's the same pattern, but outside the neuron, they can have different shapes. And then there is a lot of beautiful math going on here in which we, I mean, Rodrigo showed his one possible way of classifying these units, which is by his paramagnetic clustering. But I mean, there are several ways in which you can do this, but in the end, you get, say, the different wave shapes, and you can get back and say, oh, say here, neuron A spiked here, here, and here, neuron B there, there, and there, and so on. So this just to illustrate, there are several ways of doing this. I mean, this was already mentioned. You can play around with the width, the high, or play around with principal components. Okay, this we saw already. Okay, so but this, again, there is a lot of beautiful math going on here. You can spend your whole life tackling this problem. There is a lot of things still not solved, right, in which you can, I mean, for instance, when a neuron bursts, it usually tends to change the action potential shape, right? I mean, the first spike in the burst may be different from the last spike, and then you can erroneously classify as another neuron. But anyway, you can do that. You get your neurons, and only with this tool, I mean, again, there's a lot of math which is not shown here, but only with these tools, a high-pass filter, localize our neurons, play around with spike clustering. Only with that, we can learn a lot about the brain. So example, so this is textbook neuroscience, right? So a monkey brain recording from the prefrontal cortex. So here's a task. There is a reward here. There is no reward here. And then for a while, the monkey can't see anything. These things are covered. And after that, he has to choose and goes there and pick the reward because it kept. So it's believed that during this delay period here, the monkey kept in his memory that the reward was there, right? So it's a classical result. I mean, there are some neurons in the prefrontal cortex that, so, okay, here we have time, and this is spiking rate. I mean, all that come from recording, high-pass filtering, clustering, and this particular unit did this. So when the monkey kept things in memory, the spike rate increased. When there was nothing to be kept in memory, the spike rate was more or less the same. So how does the brain work? I mean, I don't know, but it seems that in the prefrontal cortex, there are neurons that seems to correlate with keeping an item in the working memory. So that's why we say, prefrontal cortex is related to working memory. So playing around with things like this. So this is a very nice, I mean, this textbook, right? This is maybe bare, I don't know. Again, so some neurons in the inferior temporal cortex that respond to face, but will not respond to other things. And again, how the brain works, I don't know, but it seems that there are neurons that have this characteristic in this particular brain region. Okay, only with tools simple like this. In the end, so I was showing one neuron by one neuron. So this is a classical tool, the way of displaying network activity. So basically X is time, and in the Y we have the different neurons. So say you put a lot of electrodes, these are the neurons you found, and each mark denotes in which time a given neuron spiked. So pretty much the end of your, I mean, you do a lot of stuff, you put the animal for do whatever task you want to record, and you get, in the end, you get this a barcode like this. And then, okay, so okay, you are a very smart guy, here's the barcode, here's what the animal was doing, and now tell me how the brain works, right? So this is one of the big problems in neuroscience. We want to know, so what's the neural code? And one of the few axioms that we have is that we believe neurons code things, right? With the neuron doctrine, we are still living the neuron doctrine. Okay. So I'm going to give an example of a classical result. So suppose you have a mouse here wandering around in an open field. Okay, this one, it's maybe, suppose there are some wires here and this animal is being recorded, right? And this is the barcode. And never mind the colors here, pretend they are all the same color. So here we have time, here is the different units. And okay, with smart guy you collected, the animal was just wandering around, and here is the barcode. Tell me how does the brain work? So it took a lot of years until they did a very simple thing in programming, right? And then decided to program for each neuron. So say whatever, let me get this neuron. I'll plot what's the spiking rate of this neuron as a function of space, as a function of where the animal is in this arena. And when they did this, when John O'Keefe did this is back in the 71, I think it's the original paper. So what we have here, each square represents the activity of one neuron, right? And the square is the space. So we can see that this neuron here tends to spike more when the animal was here. You see? This other neuron here tends to spike more when the animal was in this part of the environment, so on and so forth. So my point being, if you were to look just at this barcode here, you wouldn't have no clue of what's going on in the brain. But if you happen to come out with the right ideas and plot the data set in the right way, you will figure out that, in fact, I mean, each neuron had information about the position of the animal in the space. Okay, so there is not big math here. It's just a matter of programming and trying and programming the right thing. So this happened in '71 by John O'Keefe, and these are the so-called place cells, right? And okay, let's see what else I put here. Yeah, then we have several examples. This would be another one, whatever. It's just the gray is the animal wandering around. These dots denote the spikes. And then you can normalize by occupancy, so how much time the animal spent in a given space bin, and that gives a measure of time. You divide by the number of spikes, and then you come out with the place fields. Anyway, some very basic programming, you get to these nice things, other place fields. Okay, I'm going to rush because I have more stuff to tell you. And the idea is that we have the different neurons. They would be a sort of GPS, right? I mean, we have different neurons coding for different regions of the space, and people play around. So suppose here you have an arena, you should have a poster here and a bench here, and they start playing around about changing like proximal cues to the animal versus changing something very far away, and some neurons change, some others don't. There is a whole lot of research going on with this. I'm going to show you a video of one of these place cells, but here, I mean, this is a very common plot. Okay, we just use different colors to denote the activity of different neurons, and the idea is that the environment is covered by all the place cells. So just to give you an idea of, in real time, how the place cells work. Where is my video here? So this is from MIT, Matt Wilson lab. So basically here we have some clustering. I don't know what he used exactly, but the different colors are the different neurons, right? And while the animal is walking, so this picture here is going to be reconstructed here. So here we have the blue neuron spiking, so it prefers this one. And we have information about space. I don't know what exactly Matt Wilson used to separate the units. But you see that sometimes the separation is not as clear, right? I mean, how do I know this is red, this is not this guy? And you see that some cells they have more than one place field. Okay. But this, I mean, of course, this is done a posteriori, right? First, you record a signal, you do all the data treatment, you get to the clusters, and then you go around, play again, and then you do it. This is, of course, is not real time, right? Okay. Ish. This is the other thing I will talk to you. Keeping on, so place cells. Just with very simple tools. Okay, I'm going to rush this. And then the same question again. So how does the brain work? Well, electrophysiology is good for us understanding how the brain works. Again, I don't know how it works exactly, but we know that in the hippocampus, we have these cells that they seem to track spatial navigation, right? They seem to inform you about the environment. So electrophysiology starts giving you a lot of insights about this, right? This was 71. And in 2005, so we are talking about a lot later, right? 34 years later, they discovered the so-called grid cells, right? I mean, this was a boom a while ago. Now I think people are more used to grid cells. But grid cells, they remind place cells. They look like place cells. The difference that they occur in the entorhinal cortex. And here's the same thing. So this the trajectory of the animal, every spike shown here. And when you do this place field-like plots, but then you get this formation of a grid. So this would be cells that they spike in this hexagonal grid. So this, you see, it took 34 years from place cells to grid cells, and mainly because I believe it's not that easy to record from the entorhinal cortex because it's very near the bone, right? And these are now spatial autocorrelograms that they show this very beautiful pattern. And the entorhinal cortex sends projections to the hippocampus, and actually hippocampus sends it back. And for those that like modeling, there is a lot of things going on, a lot of models, very nice. I mean, again, I don't know what's fantasy, what's for real, but a lot of fun models trying to figure out how grid cells and hippocampus they can interact to anyway, how one gives can contribute to these patterns of activity, right? Okay, I don't need to go there. I'm crazy. I can use this. Okay. And now, so the grid cells, they very simple tools again. And again, another very simple tool. They recently discovered the time cell. So this was the first paper showing time cells. Whoops, where am I? And 2008, and then McDonald's neuron, who knows, it's like 3 years ago, right? Anyway, so this is a very hot topic now because, I mean, people know, okay, we have this evidence from the rodent literature showing that the hippocampus is important for spatial navigation, but we also have classical results showing that the hippocampus is important for memory. And actually, in the animal as well, is important for memory. And people, I mean, I don't buy this story, right? But now, I mean, for those that like modeling and all that stuff, now, so we have cells that mark the space, and now discover cells that mark time. And then we have all these spatial temporal things that you can play around and create fancy theories. But anyway, so what are the time cells? See, all this happening right now. I mean, so basically here, the task here, it's a classical T-maze, right? So the animal stands in this running wheel for a while, then it goes down and it has to either go right or left. It's an alternation, right? So one trial goes right and it stays here for a while, running the wheel, and then here goes left. So the animal has to alternate. What we have here is the place cells they found, okay, in different colors. Okay, so far so good. But they also discovered that during this period where the animal was here, and in theory the animal should be keeping in memory, so he should be keeping in memory that he came from this path, and that the animal knows that he has to go to the other path. So during this period here, there were some neurons that were active. They would not be active here, but they would be active just in this, say, inter-trial interval, in this delay period. Whoops, I don't know how I did this. What's going on? Okay. So here is the period inside this running wheel. So we have time in the x, and here is not like different neurons. So each rectangle like this denotes the same cell. So the same cell plotted in several trials. And the result here is that, so the animal got inside the wheel, so this cell here would always spike when, whatever, 2 seconds has passed. It's in different trials. This other neuron here, whenever, whatever, 4 seconds have passed, the neuron would spike in different trials. So this is quite very remarkable, right? I mean, it's a whole different trial, and the cell spikes at 5 and whatever, 13. I should say that, I mean, it doesn't go more than 20 seconds. So that's why some people don't buy the idea that these guys are really representing time. But anyway, so these are the time cells. And also they found this thing here. Some time cells, so there is this conjunction of coding. Some time cells, they would code, so this one would code for whatever, 3 seconds inside the wheel in different trials, but they would only spike for the 3 seconds when the animal would later choose the right choice, right, would turn right. This other one would spike for whatever, 0.5 seconds, but only when the animal would go left. And this is the next video I'm gonna show you. So this is from Bujak lab, where I have my videos here. Okay. I seem to be stuck. Okay. Ideas, reset, plug it out, plug it in. Jesus Christ. Sorry, guys. What's that? Keep asking. Oh, great. I don't know what I did, but it worked. So here we have the animal. So this is the wheel, right? We are only going to see the LED. And in the slide here, the animal was going down and it had to go left or right. But here in the movie, the animal goes here and then has to choose right or left, but it's going to be up or down, right? So this cell spiked. And then the animal went left, right? It went down, whatever, left. Here. It's an alternation. Now this animal has to go, whatever, right or up. And the cell didn't spike, and the animal went up there. See? And then it should not spike, and the animal should go up. Something interesting is going to happen, so I'm going to keep showing you this. So it spiked, then down. The problem is working with animals like that, right? I mean, they're not the smartest. And now it should go up, right? Come on. You see, it should go up, but the cell spiked. Come on, damn animal. You see, it's just a thing. I mean, the animal made an error, and the cell kind of predicted the error, right? So that's this should have been up, no? And then now keep alternating, go up. It's almost finished. There is this last one is interesting as well. So the animal should go down here, and it spikes. It made a mistake. Kind of the cell reminds the animal, okay, go down, and the animal goes. Okay, so these are the time cells, but they are not only time cells, you see. And then, I mean, there is a lot of works in the percentage of time cells that can be place cells. There's a lot. This is a very hot topic in the hippocampus field these days. Okay, meanwhile, using the same thing of just doing spike clustering, what people have been finding in humans. So this is a work that actually has to do a lot. Rodrigo is not in this paper, but has to do a lot of Rodrigo's work, like tracking these neurons that would be somewhat grandmothers. I mean, I'm going to show you. Hold on. I hope this thing doesn't get stuck again. No, I have mine. Okay, here we have. So these are epileptic patients that do go. Is this playing? Yeah. So they have electrodes inside their temporal lobe. I mean, they're being mapped for brain surgery because they are chronically intractable epileptic patients. It's not with the. That's crazy. Yeah, we are missing something here because this guy should be playing as well. But the thing is, so this is a human being. Whenever he watches the cartoon of Simpsons, the neuron likes. See, it just spikes for the Simpsons. But the weird thing is that we should have a sound for Martin Luther King and for all. I don't know what's not playing the sound. So the patient is watching this movie, right? So we saw this very specific cell that, I mean, this is firing rate, this is time, that only spiked for the Simpson. It's crazy. I mean, there is sound here. Should be. I don't know what's going on. And then we can predict what scene is gonna see. So it's the Simpson neuron. But the nice thing is, so this was, say, during encoding, right? The patient was watching this movie. And later on, the patient was asked to tell, I mean, tell me what movies do you remember seeing, right? Pre-recalling the episodes. And now, so the patient is not seeing anything. He's just saying what he saw. Oh, so remember the stock market there, right? Hollywood. So see, even this predicts. It starts, and then the guy says The Simpsons. So what's amazing is that we do believe this, right? We do believe that neurons code information. That, but this is an actual proof for that, right? So the neuron spiked not only when the patient saw The Simpsons, but also when he recalled seeing The Simpsons. So this is the way that goes in the direction that we want things to go, but I mean, believing the way things should behave and actually behaving like that is, I mean, I think that's a very remarkable result. Okay, and again, just playing around with finding, we're having very well isolated units. So actually, this is the neural. So here we have different trials. So this is time, different trials. So the different scenes, right? And here is the spikes and the amount of spike integrated here. So the neuron we saw is this one here. Actually, I also liked a little bit this Seinfeld. But anyway, this is very interesting. In terms of mathematical tools, nothing, right? This is high school. I mean, you just put the spikes here and you count the spikes. So this is period stimulus time histograms. And I mean, you can slide a window integrating spikes and have this. Okay, so, and then we get back to, we saw just a couple examples. So how's the neural code, right? Because again, we have these bar graphs. So now we know that there are whatever neurons that they can spike in a given region of the space. We have grid cells. And on the other hand, we have these very similarly specialized neurons that are very specific, right, for some features. Just to put in the plate, I mean, we know about the turning curve, right? So this is Huber and Wiesel, the classical papers, right, the Nobel Prize. So basically, this is, I mean, this holds for several things, holds for monkeys. And but pretty much in V1, we have neurons that would respond depending on the orientation of the bar that is being presented in their receptive field, right? So here we have the bar is rotating, here is the response. So this particular neuron would spike when the bar is vertical. This happens in sensory systems, this also happens in motor systems, but the idea of this turning curve. So this is a particular neuron, I think you saw this figure before as well. So basically, there is a given orientation they prefer, they spike more than to the others, but the idea is like the different neurons, they prefer different orientations. In that way, you can kind of I wouldn't say construct the image back, but have an idea of what's, I mean, at least for this simple stimuli, have one idea of what has been presented. This also happens for the motor cortex. So another classical result, but here is, I don't know if this is M1 or some supplementary motor area. But the monkey has to do these movements here. See, you have these different directions, so we have several trials here. So we have the classical rasterogram. And this cell tends to spike more when the animal is to turn to the left. You see, and when it goes right, it not only it doesn't increase the firing rate, but also decrease it. So, and again, we can have this turning curve of how the neurons are encoding this movement. And you can play around, and you can actually, so if you have the activity of several neurons, so each neuron has a preferred direction, and you can make a sort of linear sum. You attribute weights for the different neurons, and you can, in the end, have a resultant vector that's gonna more or less, so this is the actual direction, and this is the reconstructed direction, right? The thing is, we can play around with this kind of reconstruction, and then this is the idea behind brain-machine interface, right? So how can we collect the activity of neurons and being able to decode these things, and well, for the case of brain-machine interface, do some movements with robots, or say. And this still keeps, this is a forever going discussion, right? I don't buy these grandmother cells, but we have some very impressive examples, right? But the thing is, to what point the neural code is centralized, like we would have someone that represents my grandmother, and if this neuron dies, I don't know who's my grandmother anymore. Or to what point, for whatever thing in the brain, we need the activity of several brain cells, and we need to decode a lot of them and play around with fancy algorithms to try to figure out what the brain is doing, right? So I don't really believe there are true grandmother cells, but there are some very impressive examples, like the work of Rodrigo Quiroga as well has been shown. But irrespective of that, in the how the coding, so this is what I call, so this is Nicolili's work, this is what I call the applied neuroscience, right? I mean, irrespective of how the coding really happens, maybe sometimes you can bypass what the neurons are actually doing and use the neuroactivity to do stuff. So in the case of Nicolili's, he trained some artificial neural networks. So there are several algorithms that can learn to do this pattern classification. So the monkey is moving a joystick, he has to play some video game. And while the monkey is moving the joystick, they record the activity here, the rasterograms, and they make a computational algorithm to learn that, so whatever, when the animals turn with the joystick right or left, this particular activity was here. Eventually, they make what's gonna, I mean, the animal is gonna keep doing the movement with the joystick, but what's going to change the actual video game is going to be the decoding of the neuronal activities, no longer be the joystick, right? And the remarkable result of this particular paper is that eventually the animal figures out that he doesn't have to move the joystick for the target to move there, right? And then there is a very nice video showing that anyway, the animal stops moving the joystick and he keeps playing just by thinking, right? So this is Nicolillis and others have been showing this. So irrespective of how the neural coding really is, we can play around with some heavy math. I mean, not that heavy actually, and do this reconstruction of things. Okay, so this was it for introduction of neurons. You guys don't worry that I will stop at 4, right? If I don't have time to see everything. So now talking about local field potentials, and so we are here, right? I just showed some very simple example, no fancy math whatsoever, just playing around with spike times. We can do a lot of stuff with the rastograms. And now let's see what kind of insights we can have looking at the local field potential. So the idea here, I mean, this would be, I mean, nothing, once you have a digital signal, you don't have, nothing's continuous anymore, right? But in terms of, in a practical way, we neuroscientists, or I at least, we think that we would have these two signals. One is the discrete, the bunch of zeros and one, I mean, it spikes, even it spiked or didn't spike. And the other one would be this continuous series, I mean, up to the sampling rate, right? So we can play around. I'm using local field potential for local field potential, but I mean, the kind of analysis you can apply for EEGs as well. I mean, the definition of the local field potential depends on the size of your recording electrode as well. To what point you are, whatever, going macroscopic or mesoscopic. So this is a very simple, not simple, but a very standard tool. So the first tool that people apply to the signals is called the Fourier analysis. So the idea here is that whatever signal you have, so there is a theorem. Actually, there is a letter from Fourier when he was 25. He was writing someone saying he was concerned that at this age, Newton already did a lot of things. He was already an immortal, and he was worried about being famous. But in the end, he got his immortality. I mean, he's dead, right? But I like the Woody Allen saying, right? I would like to be immortal by not dying, no, not because of my work, right? But he's one of the immortals out there, even though he's dead. But so whatever signal you have can be represented. I mean, a stationary signal. There is a lot of technicalities here, but let's pretend they don't exist. So whatever signal you have, it can be represented by a sum of sine waves of different frequencies. Okay, there is a theorem out there. You can think of this as there is a space, the same thing as we have coordinates, right? We have R3, R2, you have a given point in a given coordinate, so a point can be represented by whatever, the value in the y-axis and the x-axis. A function in this infinite space can be represented as a point, and each coordinate is going to represent the amount of the amplitude of the frequency that is represented in that axis. So in math, this is called functional analysis. But the idea for us here, very, simple stuff. So pretty much, people talk going from the domain of time to the domain of frequency, right? But suppose here you have a sine wave of 80 hertz. You apply a Fourier transform, and then actually you have to, the Fourier transform by itself is a complex number, but you have to compute the length of your Fourier transform. And this is the so-called power spectrum. It basically tells you for the different frequencies, the amount of you had for different frequencies. So in this case, we only had 8 hertz, and it shows up here, peaking 8 hertz, okay? 4 hertz, same thing. This is 50 hertz, same thing. It's just noise, white noise. It has all frequencies, right? And my point, so this is, of course, fake Robocop signals, MATLAB stuff. When you sum everyone, you get this. It's a little bit harder to see, but nevertheless, when you do this frequency decomposition, this Fourier tools, you're going to capture the peaks there. Okay, it's simple like that. So this is very used. There is a lot. There's a lot of stuff behind this. When I say simple. And the results here is that. So here you have your. Where is this from? This is locus. I think this is Gilles Doran, locus. Factor bow. But the thing is, there is an odor that's coming on here. We have 4 recordings, and this thing shows up. You see, when people go to the lay TV and say, brain waves, these brain waves, they're not lying. Brain waves, they do exist. So here we have an example of a brain wave. So there is an odor here, and in these recordings, they start becoming oscillatory. And then you can do, in this case, it's so beautiful, you don't even need to do these Fourier things, right? It shows up here very nicely. But my point being, for those that haven't seen, their brain waves, they do exist, right? When people talk to you that they study brain waves, they're not crazy. These waves do exist. And of course, we like to believe, there is a lot of debate as well, we like to believe that these brain waves, they would have, they would be doing something for brain computation, right? I mean, because this is a sensory and the olfactory bulb, we are having odors here, and why this guy would show up? And so this is the field I work, I mean, not with the locus, but I work studying these brain waves. This is a paper with me and cats. I don't know if it's still around, but basically here, so cats talk to you. So he has the model with an intraoral cannula. He delivers fluids over the tongue, and he records from the gustatory cortex. And what we saw here, so 0 is the period when the fluid touches the tongue, and this, so here it's frequency, here is time. So basically I'm computing the power spectrum that tool will learn, but in several windows. And I'm sliding these windows and I'm plotting in this color mode. And the result here is that this wave, there is an evoked response that gives rise to all this power here in this low frequency, and then so whatever, 8, 9 hertz it's up here, and then it slows down. And then we get back to the question, how does the brain work? I don't know, we are trying to figure out, but I mean, as another empirical evidence, I know that when the tongue touches your tongue, when the taste touches your tongue, there is this wave that shows up in this cortex here. What this wave is doing? What generates this wave? Well, I mean, there are several further questions, but this would be a first empirical evidence that I like to believe that these brain waves are doing something, but who knows? I told you this as, I mean, there is the notations are the different Greek letters, and actually this varies from lab to lab, and varies depending on the the species you're working. I mean, theta in rodents can be up to more than 10 hertz. In the humans, 4 to 8. So it's somewhat messy. Okay, there is no fixed definition. I mean, the boundaries are not as fixed depending. And actually, I'm one that promotes a lot that we should stop defining brain waves based on frequency range, because we know very well that if you take what we call as alpha, depending on if you are in the hippocampus, alpha is actually theta. If you are recording the spindles, alpha are spindles. I mean, it makes no sense to my mind to keep calling the oscillations just based on frequency. And people go there and they start discussing, you get a paper on humans and they found something that happened in theta, and then they cite the rat literature. I think it's a big mess. And I advocate that forget about its frequency range definition, because for the same frequency range you can have very different things, I believe. But anyway, but this is the way it is right now, right? You have these different ways of calling it. So this is a very classical wave in the hippocampus. This is theta. And I mean, this is one of the few exact things we have. One of the things, this looks like math, because whenever the animal is walking, wandering, the theta waves will show up, right? If the theta waves don't show up when your animal, when your rat is walking, that means that you are not in the hippocampus, right? So some things are very, very robust in neuroscience. The appearance of theta waves during exploration in rodents, this is one of them. And then you can do the Fourier spectrum, and there you have your theta peak. Well, there is a lot of gamma stuff going on. I'm going to get back to gamma, and I should just say, I mean, I'm very skeptical. So there is literature that will show you whatever you want, but I mean, gamma seems to show up during memory, during attention, but there's a lot of debate going on. But it's good, again, to be on this field just so you can put your gloves and come fight. Come to the skeptical side and let's stop the fairy tale. And then we can play around with the waves and how the units behave. So we know, so this can be a theta wave in the hippocampus. Okay, this is just a sine wave. But the thing is, if you look at the probability of your neuron emitting a spike, you're going to see that there are higher chances that your neuron is firing depending on the phase of the theta, right? Depending on whether it's at the trough, at the peak, rising or descending phase. So we know that the local field potential waves, they modulate the activity of the units. And some people like to think that, so timing, for example, right? We know that the brain uses time, but how is time encoded in the brain? So one of the theories says that maybe these waves, they could be a sort of a clock for the neuronal activity to be organized. Coming back to, John O'Keefe is the guy that discovered the place cells in 71. So in 1993, we are talking about 22 years later. In 22 years later, he decided to plot. So let's go little by little here. So here is a rat going a linear track, and this is the place field. So this particular cell spikes when the animal is here. See, we have the place field here. And so he's just plotting the cell here, the spikes of the cell as a function of time. And here is the LFP. And what he did then is to see, I mean, you can filter and see at which phase this spike occurred. Okay, this one maybe is a bit messy. This one is easier to explain. So here is space. So as the animal goes, and here's the phase of theta. What we see is that we have this precession, you see? When the animal enters the field, this spikes more at this whatever phase, and while the animal goes through the field, the phase changes. And then people start discussing. So it's not so. Place cells, we know that they have what we call the rate coding, right? They increase their spiking rate to denote a given position of the space. But if you think, I mean, we can also decode the position based on where the spikes are occurring, depending on the phase. Because here you would have, say, a bell-shaped curve. So the firing rate would increase and decrease. So for a given firing rate, say here, the animal could be either leaving the place field or here could get in the place field. So firing rate, the same firing rate occurs in different portions of the field. And some people say, oh, we can disambiguate that if we look at what phase the spikes are occurring. So this is also theory out there, but the thing is, maybe the coding is not only rate, it's not only increasing in firing rate, but it matters the precise time in which the spike occurred, or in this case, timing measured by phase. So it will matter the precise phase in which spikes occurred. Okay? Same guy, it took 22 years to discover. This is a very robust effect. It took 22 years to discover phase precession. And why? I mean, he had the way of doing that before. Because of a matter of writing 2-3 lines of MATLAB and I don't know MATLAB, but it's just a matter of programming the right thing, and then you're going to find it. You know, it's just a matter of programming the right thing. Because he had never before got the phase here and programmed this kind of analysis. So in short, keep your data. You never know when you're going to figure out something there. And nowadays we have these theories about how the brain waves, they could be useful for communication. The idea is that they would lead to ways of depolarization and hyperpolarization inside the cell. Anyway, and the fact that if you have, so these would be different brain regions, and if a given region was synchronous with another up to the delay of the conduction delay of the axon, they would whatever exchange information better than if the regions, if the brain waves in a given region is not phase locked with the other one. So this is theory, right? There is Pascal Fries, one of the main guys that behind this theory. There are more people, but I'll put my mystical sign here that, well, it's still debatable. But these are the theories we have out there. So I'm going to mention another measure that's very standard stuff. It's coherence. Coherence is simply a measure of phase lock, and there are actually several ways of view for measuring coherence. I'm going to just show you the Fourier standard coherence. It's something that varies between 0 and 1. There's more here, there's more, but I'm just going to talk about one. But the thing is, coherence measures how, so it's between two signals. Before, power spectrum would apply to a single signal, right? Coherence, you need two signals and then to compare. And basically, so in this example, so we have, say here, whatever, I would have the hippocampus, this prefrontal cortex, just lying here, but just for and the thing is, when we overlay both, we see that whatever, the peak goes with the peak, the trough of the trough, they have the same phase for most of the part, okay? So these two signals, they are said to be coherent. They don't have to have this zero phase lag coherence. In this case, they have, right? But the thing is, the important thing is to keep the phase difference constant, phase difference. And here's an example in which, if I overlay, I put one above the other, you see here we have the trough of one going with the peak of the other, and they don't keep this, because here peak goes with peak. So there is no systematic relationship between these two signals in terms of phase locking. So these two signals, they are said to be non-coherent. I'm going to show you some results with this coherence analysis. So this is again with Matt Wilson and Matt Jones. So here is the typical decision making for rats. So basically the animal goes here, this is a kind of a wall, so the animal has to turn here, and here it has a choice to do, it has to go wherever, left or right. F means forced, so the animal here is forced to come here, and here, whatever, has the choice of choosing between going up or going down. And then you would have this called forced returns or forced runs, in which the animal, they had no choice, so the walls were here and here, the animal would just get back. Okay, so there is choice in this period, there is no choice in this other trial. And here, one is the hippocampus, the other the prefrontal cortex. And so the idea here, the result they found is that when the animal has to choose, there will be a higher coherence. So this is the amount of coherence between both these regions, then compared to here when the animal, he doesn't, in theory, doesn't have to think, just have to follow the walls here. So this as power, we also have a spectrum for coherence. So this would be the choice runs, this would be the force runs, and then you play around and you show specifics for theta. If you go at delta, which is the name of this band here, you don't see the difference. And here you would see that there is a higher coherence between both these regions when the animal has to make a choice. And actually this is even it's better than that. It not only has to make a choice, but a correct choice. Wrong choice would be associated with a lower level of coherence. And this gray here would be forced turn. And again, I mean, are brain waves important or not? I mean, this kind of course, this has to be. Actually, this was replicated by Joshua Gordon, if I'm not mistaken. But believing this result holds true and it's replicable, and then we have to, okay, so what brain waves are doing? So maybe they're indeed helping to, whatever, the hippocampus talk to the prefrontal cortex during decision making, because we have this very nice increase in coherence there. This is the reference. There is also a result regarding coherence. Here is also TMAs, this is again Bujaki lab. And again, left or right, and they are plotting, it's a, so programming, do all your Fourier stuff, get your coherence estimate, integrate the coherence in the gamma band, and then you plot here as a function of space. And he found that the coherence between these two subregions of the hippocampus tend to be higher in the long arm. Actually, I have a video for this. You're going to see that in the single trial level, we can see this coherence, but it's not like clear water. Like as everything in neuroscience, actually this is a problem in neuroscience because oftentimes we report results that come from averaging along several trials, and then very neatly things shows up very nice, right? But the brain has a single trial, right? When you have to whatever escape the lion is that trial, right? You don't average several things for that. But anyway, in the average, this looks very nice. In the single trial, it's still nice. I mean, up to being biology, right? I think it's this one. So the animal is going. So this is coherence. This is the hippocampus. But so this is the reference. This is CA3. So this is CA1. I see when it was here, you should have seen the red shows up here. But the thing is, I mean, some red shows up here and there, but when the animal is here, there is more red, meaning there is a higher coherence between the CA3 and the CA1, right? Again, when the animal passes here, you should see some red here. This is the result, you see? This red here. But again, notice that in the single trial, I mean, it's noisy a little bit, but I mean, and when you average, you get that really nice picture. I mean, that one was a function of space. He also has average for these spatial maps here. Again, the animal is going, and you should see, where is it? Come on. This red here that shows up. And this is math, this is programming, right? Even though there is not a single equation here, but there is, I mean, you collect the data, low pass filter, and then you play around with your this is another stuff, sorry. Actually, I'm not showing this. And you play around with your Fourier and get your coherence estimates for several things, and then plot as a function of space, and from there you go. Okay. Maybe I should skip this one. Yeah, you know what? I will skip this one because we are not talking about simulations here. Maybe I should go to part 2 and part 3. Yeah, so no computational neuroscience. Let's stay with the data analysis. Let me see here. 1 second. Okay, no. I'll stop here and I'll jump to, so this was the nice videos, now it's my dull work, right? So, yeah, forget about computational neuroscience. Let me go here. So I'm gonna show you one tool we came out to analyze the units, and then, if time allows, I'm gonna show another tool we did for analyzing the LFP. So now it's our own research. So again, so this first work is done with. Okay, so now we are here. I just jumped the computational neuroscience part. Now we are here. Okay, so we are talking now. Okay, we know that the analysis of spike activity. This is part of the PhD thesis of Vitor. And okay, we know about this. We are back to the neuronal units. And the idea here is, so we have this concept in neuroscience, which was created by Hebb. And I mean, putly saying is that things will not be represented by a single cell, but the activity of cell is spiking synchronously, right? So say here I have a bunch of cells, and these 5 cells here would be the ensemble or the cell assembly that would represent, I mean, it can be a sensation, it can be a motor act, it can be an idea. Okay, so we want to find cells that tend to spike together, right? And the synchronous spike would, I mean, this is the axiom, right? Would represent, well, it can represent several things, up to motor action to some sensor. Here is a sensor input. So this would be a cell assembly, and Hebb has also this idea. He went further to say that he talks about phase cycles, right? In which a sequence of cell assemblies, which is represented here. So this is the cell assembly, again, a group of neurons that fired together. They were active here, and then this other cell assembly was active here, this other one here. So this would be the phase cycle that Hebb postulated to exist. Right? And one would be calling the other. This is like, you know, when you start singing a song, and then the rest of the lyrics just comes with different cell assemblies calling each other. Okay, there's a lot of theory here. A lot of people believe in cell assemblies. I think I do believe in cell assemblies. But we have yet to prove that they do exist. I mean, in a very definitive proof. Okay. Okay, this is our other work talking about. If the cell assemblies, they can be. I mean, you can have a cell assembly that represents whatever represents the IMIRA. And the idea is that it doesn't have to be restricted to a given region. You can have cells in different parts of your brain that when they would fire together, they would represent this assembly. Or I mean, put in a very naive way, maybe I'm completely wrong, but if you think in an orange could be your neuron for the circle and your neuron for the orange color spiking together, and this is the fruit orange, whatever. This is the scary diagram that Don Katz mentioned the other day, right? Okay, just a remark then. So I'm going to show you a tool that, so there are some assumptions. And, so getting back to the story, this tool happened when, actually we read a neuron paper by Battaglia. And in this neuron paper, they use this tool that is described in this J Computational Neuroscience paper, right? I'm going to tell you the story from the beginning. But pretty much here they were using principal components to record cell assemblies. But this paper by itself was also inspired. Everything goes back to Nicolidis. This is a science paper in 1995 in which he was also using principal components to track this cell assembly. And I mean, there are several names, right? Some people just call it simply as ensemble activity. The advance brought forward by Adrian is that he came out with a statistical threshold to say to what point the assemblies really exist or not, while here they would get some principal components that they would not have a criterion to say whether assemblies were there or not. But I'm going to start from the very beginning. You guys will understand, hopefully. So this is how the framework works. Okay, so we know this. So this is the rasterogram. This is time. These are different neurons. Okay, this is just a zoom-in view of these whatever neurons from 10 to 15 here in the time. So what we do, we get the timing here, the rasterogram, and we bin time. You see this dashed line is just a time bin. And for each time bin like this, we just count the amount of spikes. Simple like that. And here it is. Okay, here is represent. So now I got this rasterogram, and now I have this binned rasterogram, and color represents my spike count. And this is just a zoom of the same period, just saying this neuron spike 0 and this being 1, and so on and so forth. Okay, this one was 3, 3. So we have this matrix here. Okay, so this is my binned spike matrix. What I do next, it's not strictly necessary though, but it's part of the framework. I just, line by line, I just compute z-scores, right? So this guy here is the same as this one here, but it's normalized, right? I take out the mean and divide by standard deviation. So now this is z-score units, right? How many standard deviation it's above or below the mean, okay? But the same one normalized. From this guy here, what I do next is this. So for example, here I'm taking the activity of neuron 16, which is here, in normalized units, and neuron 3, which is here. I then compute the correlation between them, right? So and then so this guy would be 16 is here, 3 is here. So this square here comes from this correlation here. And this is the autocorrelation matrix. Actually, this is the same as the covariance matrix, right? Because since the mean is 0 and the standard deviation is 0, I mean autocorrelation or covariance matrix in this case is the same thing. So I come out with this, and I can see that some neurons are correlated with others. So this guy is correlated with these ones, others they are not. Of course, a neuron is very correlated with itself, but we put this to be 0 just so we can see the others, because otherwise the main diagonal would be 1. Okay, so this is my autocorrelation matrix, and it's just indicating the level of correlation between different neurons. From here, I compute the eigenvalues of this matrix. Okay, so if I have whatever 20 neurons, I have 20 eigenvalues here. So this is the histogram of the eigenvalues. So to each eigenvalue, so here I have 20 dimensions. I have 20 then principal components, and here they are. Okay, first principal component up to the 20th principal component. Okay, so far so good. I get my spike, I bin, I normalize, I compute the autocorrelation matrix, and then, I mean, of course, there is a lot of linear algebra going on here, and but this is also one line of code in MATLAB. It's not programming-wise, it's not very difficult. Okay, we are here. We have the principal components, and okay, they were here at this time, and you notice that some eigenvalues they are higher than others. Okay. So what Nicolelis did by the time he would get, I don't know, maybe the first 4, the highest 5, the highest 4, to use to get back to your data and project the activity in these dimensions, right? So this is his science paper. So he has recorded from several, this is thalamus, right? From several areas, S1 maybe here. And here is so you use all these guys here, and then you use, so you know, this is a tool for reducing dimensionality. Here we have, I don't know how many neurons, but say 50 dimensions, and then in the end we get a single trace here, which is the projection in the first principal component. So what's the projection? You can think of a principal component if we get here. So this, let me get this principal component here. You see, so it's a vector, which in this case has 20 dimensions because we have 20 neurons. And it gives weight to each one of these neurons. So a projection would be, I would get, say, a column here of activity, and to get a single number, I would multiply neuron by neuron. So neuron 1 would be multiplied by this weight, summed by the weight of neuron 2, times the spiking rate of neuron 2, and so on. So this is a kind of intern product, right? So you can do this. And they did this, and they found these oscillations in the activity of the neurons when you looked at this principal component. This was somewhat replicated by Beirash. I mean, he started doing the same thing, right? So he got the first principal component, the second one. Here's the neurons they recorded, and here you can think. So they were referring to this time courses here, these projections of this reduction of dimensionality. I refer to the activity of a given assembly, right? So we have 2 assemblies here, one is active here, the other is active there. They went on to show some nice stuff. So here would be an assembly activity before. So this would be, I don't know, rectangle, maybe slow wave sleep. But before a given task, after learning something, they show, you see some ensembles were formed during the experience, which is not shown here, and then we have a higher ensemble activity here than before. And I mean, in a way of going along with Hebb's idea of assembly being formed with experience. Okay, and then we played around. So this is very, I'll show you very fake stuff and then some more realistic stuff. But we used the very fake ones so we can see it. This is a rastergram, and clearly you guys can also see there are 2 ensembles, right? Cell assemblies. Here's one, here's the other one. I mean, clearly, right? Very, I mean, you don't need fancy tools to see it. But then we did all this framework, and here's the projection in the first principal component. So it's peaking whenever this assemble is active, and the other one when the second. I mean, this is the first here, but whatever. The principal component attribute to this one to be the second one, and it's peaking here. So okay, this thing works, nice. This is a very fake example, but it's still just to illustrate. But then when we played around, and because I mean, in theory, the cell assemblies they came. A cell assembly is only representing what's meant to represent when all, or the majority of the neurons are active. But a given neuron can take part in one assembly and also in the other. So say my round neuron would take part in my orange fruit cell assembly, but can also take part in my apple cell assembly. And when we played around with, so here we created two cell assemblies, but they would share neurons. See, there are two neurons that are common to both of them. And then we saw that this method would just break, right? The principal components would not be able to separate them out, and then we have the estimation of them would be the same. So this is wrong. So we found this flaw in this methodology of computing assembly with principal component. And the work that Victor did was trying to improve this method to be able to separate out assemblies even when they share neurons. Okay. Okay. So the idea here, we used something that Peirache did, which was the improvement over Nicolilli's framework. And what he did, actually he was talking, I talked to him and he explained me, I asked him, so how you came out with the Marchenko-Pastur distribution? I mean, nowadays we use every day. But he said it was just like talking in the eye of the university with a physicist friend of his, and he just told him about this distribution. So this comes from random matrix theory. So if you have a matrix that is, the entries are Gaussian distributed, like stochastic process Gaussian distributed, and if there is no correlation among rows, the eigenvalues, they are predicted to fall. So this is the distribution of eigenvalues. Oh, this is normalized, right? So one reason why we normalize, so is things get, anyway, around 1. But the distribution of eigenvalues, they are predicted to fall within this Marchenko-Pastur, which is this guy here. Okay, so this is theoretical distribution, the formula is over here. And these are the empirical results. I mean, we simulated this a lot. And the point being, I mean, you have a greater uncertainty. I mean, the distribution is wider if you have more neurons. If you have more neurons for the same time, you have larger, and or opposite, if you keep the same number of neurons, but if you are looking at less time, then you also have a wider distribution. Then we played around because, I mean, in theory, this guy here, this distribution, again, that will tell you the probability of your eigenvalues. This was to Marchenko-Pastur. Probably these are two guys, right? They derived it for random matrix of very large matrix. In our case, our matrices, they are finite, and the entries, they're not Gaussian. But we played around numerically, right? And numerically, we could prove that this was also a very good approximation for our case, in which we had finite matrices, and the entries were not necessarily Gaussian. Okay, this I already told you. I mean, the lower the number of neurons, the larger, or the less time numbers. Sorry, the higher the number of neurons, the larger, and then the lower the number of time you have to analyze, the larger as well. And here's just playing around, but making a long story short, basically, the red one, it means that it didn't, means that all eigenvalues fell inside the Marchenko-Pastur distribution. And this will always happen. Actually, I mean, the theorem is like that. But again, the thing is, the theorem was proved for random, very large matrix. But the magical number here is that your number of beans has to be larger than your number of neurons. If you have that, the Marchenko distribution, it doesn't fail, even for empiric at least. We spent a lot of Victor's work playing around with matrices, and it worked for several kinds of things. I mean, I believe this is true, but it was, again, only proof for Gaussian process, but empirically we found this to be true for whatever other process. Okay, and so here's a very fake signal, again. One cell assembly active here and here, and then this is the autocorrelation matrix. I mean, those 3 or 4 guys here, they show up in a very nice correlation. Of course, you don't need all the framework for these fake guys, but just to show you the principle. And so here's the theoretical distribution. But now these things, they're not, the rows are not independent anymore, right? Because I have that the activity of this neuron correlates with this other one. And so this would be the distribution if there were no cell assemblies. And what happens that we have one eigenvalue. So this is the actual eigenvalues for this guy here. We have one eigenvalue above the distribution, right? And then when we program 3 assemblies here, again, very fake stuff. We see the 3 islands in the autocorrelation, and we get 3 guys above the theoretical distribution. So this was the contribution of Peirache to only get, because before Nicolilli's was getting as, I don't know, 5, 4, 6 principal components that would be their cell assemblies. But in the end, you should get, the number of principal components that you should get is the number of principal components that is above this theoretical distribution. Another nice thing is that this is analytical, right? There is a formula for this, so you don't need to do tons of surrogate analysis to see what's statistical and what's not. Okay, so now we had a very nice analytical threshold to say, in this case, 3 cell assemblies were present in our network. Here is only one. But in addition, you can see that some eigenvalues were below the border of the distribution. And here is more. It turns out that the number of eigenvalues, either above or below, will tell you the total number of neurons involved in cell assemblies. Right? Again, number of eigenvalues above the distribution will tell you the number of cell assemblies. And here is just one cell assembly, but we are increasing size. See, just one cell assembly, but with whatever, 3, I don't know how many here, but more and more neurons. There is always only one above, but you see that below we start having more and more guys. And okay, we played around, but this I should say is numerical, right? We did tons of simulations, and it turns out that so the number of eigenvalues outside the distribution will mark you the number of cells involved in the cell assemblies. These are some. I mean, we did several parametric studies. I will not just focus on the main things here. So this is our framework then that we came out to try to fix that problem of when you have a cell assembly sharing neurons among others, that the method will not work. So here is your spike matrix. Sorry, yeah, your rastergram, binary rastergram. You compute your autocorrelation, and you can see that some neurons would be correlated here. See, this is a more difficult example, but it's still easy. Because before I was putting them all in order, and now I scrambled around, right? But I mean, you can see here, and then you compute the eigenvalues, and we see that there are 2 eigenvalues above the distribution. That means there are 2 cell assemblies here. And then we just look at the first 2 principal components, and the guys actually already tell you. See, principal component 1 is telling you whatever, neuron 5, 15, and 21 is one assembly, and neurons whatever, 12 and 20-something, 23 is another assembly. So what we do then, we have two cell assemblies. So we only look at the first 2 principal components, and we plot every entry against each other. So whatever, this 23 here would be for principal component 2 would be this value here, which is this guy here, and for principal component 1 is this dimension here. And when we do that, we clearly see that some neurons, they don't have weights, and others have. Okay, from here what we do, and again, this is very easy for now, but it's going to get more complicated, but we can already see that they tend to cluster. The cell assemblies tend to cluster. What we do next, we get, and so we have the information that there are 5 cells that should pertain to cell assemblies, because we have 2 guys up here, and we have 3 guys. There is 1 in here, there are 2 together. And then what we do then, we get the 5 largest vectors in this space here. So these are called our assembly space, just to give a name. And it turns out, okay, the 5 ones with the highest length, they are here. And what we do, we compute the inner product among them, right? So these guys are roughly perpendicular to this one, would give rise to a low inner product. So this matrix here, this is what we call, just to give names, interaction matrix. It's just telling you what's the inner product between these neurons. So 5 goes well with whatever, 15 and 21, you see? And here we see 12 and 23. I mean, you can clearly see by eye here. But eventually it's going to be more difficult, and then we need this matrix here, which is just telling you the inner products among the 5 highest ones. Okay, these are the 2 objects that we have. And what we do from here, we make this guy, we come out of a threshold and make this either 0 or 1, and then we have a clustering algorithm that will, so in this case, again, we don't need any math, right? You just look and see who are the cell assemblies, but it's going to get more hairy than this. But we came out with, there is an algorithm that I can cluster which neuron pertains to which. To be honest, the algorithm is quite difficult. This was done by another author in this paper, which is Sergio Conde. But when I was writing this paper, I said, at this point we use a simple clustering algorithm to clusterize the assemblies and see supplementary material. And to these days, I don't know how the algorithm works very well, but I call it a simple clustering algorithm. It's there. You guys want to try it? It works, because I tried several matrices and it worked. Okay, and this is done, and then we get our cell assembly neurons. This I told you, so this is the interaction matrix. In the end, we let it binary, either 0 or 1. But okay, let's get back here. And once we have the assemblies, what we do, we reorder our rastergram, and we also come out with what we call, so this method for identifying cell assemblies is what we call the assembly vector method. So for the guys that pertain to the same assembly, we compute the mean vector, right? It normalized to be 1. So this is the assembly vector for this guy. This is the assembly vector for this other guy, which is the average vector. I mean, the average direction is normalized to be 1. And then we construct, in the same way, Pirashi and Nicolidis were projecting stuff using principal components. Now we are projecting stuff using a linear combination of principal components. And the weight of this linear combination is nothing else than the coordinates here of these assembly vectors. And then the projection is the same, and we go there and we see the two assemblies here. So this is one assembly, this is the other assembly. Okay, this case was easy, but then we can get the case in which we have mixed assemblies sharing more than one neuron. So here we have 3 assemblies, we have more guy here, and then we look at the first 3 principal components. These are our assembly space. Now it's a little bit more hairy to classify things, but we can, so this is the interaction matrix, and then we apply our simple clustering algorithm and gives you magically the guys. And then we reorder things, and now we see that some assemblies share neurons, and the rest is we compute the assembly vectors. So now you see, if we were to apply the original framework, I would use as projectors of the activity the principal components. And here we see that the assembly vectors, they are linear combination of the principal components, but they're not the principal components. We create the projectors, and then we plot around, and then we have the activity. So we have the time course of the activity of these 3 assemblies. And just to make you the case, this is using principal components. Okay, in this case, it would give you the same result, except that what's called as 1, or 1 is here, whatever. But for the case of the mixed assembly, it would mix up. You see, the thing would not work here. So we are pretty happy by the time. And then we end by publishing the methods. And this first paper was more of a methodological one. Okay, so now we can track activity of cell assemblies even though they share neurons. So we don't have this flaw anymore. And then we spend 1 year and 1.5 years working on a paper. And I should say I will only show 2 or 3 slides of applications, because then we didn't get confident anymore that the thing is, the framework will work if you go back to the very beginning, if your spike clustering was well done, right? And here in this paper, this was part of Victor's PhD, we found very nice assemblies that would oscillate when the animal was seeing some, exploring some objects. But in the end, looking at the videos, we saw that at the same time, the animal tend to choose something, right? It might be the food, might be, in the end, I don't know, we didn't get that many. Anyway, there are the applications, we just put it out. So, but just to say, I mean, the method doesn't do any miracle. Before that, you need to have a very high quality results, right? But it's still, so this is actual data. We can still find assemblies and play around and show that the assemblies there. So these are different brain regions. You can play around with the plotting their time course. So they do exist. Here's just to show that this is our method. This is the original method, and indeed they give rise to different results. This guy tends to mix things that are separated in this other one here. I mean, again, at this point, as applications, I would show your paper, but I mean, since we don't believe it anymore, I just put it away. Here is the actual data, and just to show you that indeed we tend to, it's not this thing to be of being bimodal, right? Some neurons having a higher inner product than the others. We can kind of see that this thresholding of interaction matrix in real data is not something that fake. And this is something I stopped doing, but I had access to some brain slices, right? And here's whatever cell assemblies in brain slices. And again, we could see, so this interaction matrix, indeed, I mean, there seems to be some neurons are more correlated than others. Yeah, so for application, anyway, we have yet to do a more neurophysiological paper of this. So far, this is more of a method stuff. So this would be the advantage and blah, blah. So this is the paper. And I should say that after this paper, as often happens, we came out with an even simpler and easier way and actually more powerful to track assemblies. I'm not going to tell you this here. But so this is our second paper in this field where, I mean, now we are going to independent component. And actually, it's a mix of principal components and independent components. And here, Siddhartha started doing with his PhD student to get this framework and go forward and start looking at the phase sequence, right? So now we are, now that we can track our assemblies, maybe we can also start working with phase sequence and see if they exist, right? The phase cycles, the things that have postulate. Now we can check whether it's true or not. What? Yes. The histogram with the 2D plots, they are linear, like this guy here. You tell me when I stop. Yeah, this interaction matrix. Oh, mhm. They have to be symmetric, right? The thing is, I mean, this method, there is also a flaw which I put under the rug, which is when we compute the assembly vectors, we only use neurons that are exclusive for that assembly. You know, so this is something I need to tell. And this is actually which we fixed in the this guy here. This guy, you're right. So maybe it's what I call an inner product. It's not an inner product. I think it's the length in the direction of the. Hold on. Here. So maybe I lied to. What I called as an inner product. Maybe it's just the length in the direction of the other guy, right? It's not in the. It's not. Yeah. It's not a true inner product. It's. Where is it? So what we do here, say the interaction between, but shouldn't, oh, that's why it's not symmetric, okay. What I call as an inner, so this is our assembly space, and I told you guys that the interaction matrix would be, say, 5 with 15 would be the inner product between these two, right? But actually, I lied, is the inner product between 5 and this, in this direction here. So this guy here would turn into a unitary vector, right? And the interaction between 15 and 5 is 15 against a vector which has the direction of 5 but is unitary. And I think that explains why it's not symmetrical. And why did we do that and not... Yeah, but this is the ones I had slides for. But now yeah, but this is how the story started, and then we figure out these things, or Victor figure out, and now, so in this other one we fixed, I mean, it's getting improved, but it's still, we assume linear stuff, right? Which is may not be how assemblies behave. Okay, last part. I know you are tired, I'm also tired, but blame whoever wants to blame. Let's go part 3. Okay, and okay. So now I'm going to, it's going to be the same thing, but here I'm going to have more applications. This assembly story, we were very methodological. We still have to find a utility, right? But we had a paper that we lost after 1 year and a half. But it was very beautiful. Well, so I'm here talking about a tool we came out to measure phase amplitude coupling, so the analysis of LFPs. And I probably won't go through all this. I'll be talking until 4, right? So, but this I will tell you 2 works I did during my postdoc, and then I will eventually stop because it's going to be 4. And then, but I keep doing this line of research, looking at. So basically, we are back to the brain rhythms, and now we are looking at how brain waves of different frequencies talk to each other. There are several ways in which this interaction can occur. So for instance, here we would have a slower wave, which I call theta. So this is my theta wave. This is the amplitude of theta, but never mind for now. But this is theta, and here, say I have a faster wave. Just to give a name, this can be whatever, beta. Okay? So there are several examples in which, say, here the amplitude of theta is changing, and the amplitude of gamma is also changing. So this is called amplitude-amplitude coupling. There are some types of coupling, but the one I work with is this kind here. So this is called phase amplitude or phase power or phase energy. In a practical way, for me, power, energy, and amplitude, they mean the same thing from a practical perspective. But the thing is, the amplitude of the gamma here increases and decreases, you see, and it does so in a way that is locked to the phase of theta. By phase, I simply mean, in this case, when theta is at the peak, we have the gamma is at the highest amplitude, and vice versa. At the trough, we have the lowest amplitude. Notice that we have a common signal here, and I band pass filtered in parallel, right? So to get this guy and to get this guy, but these two guys, they come from a same LFP. So how do we measure these? This, I will start with fake signal. So this is the Robocop LFP. So I hope you can all see we have theta here. And riding on this theta, we have the faster, the gamma here as well, right? So, and I hope you can all see that the amplitude of the gamma is highest here when theta as well, in this example, when theta is at the peak. And when theta is at the trough, we have a lower amplitude gamma. Okay, so what we do, so this is the framework for measuring phase amplitude coupling. I filter to get the theta, it's here. And I also get the same signal and filter here to get the gamma. The amplitude of the gamma is lower than the theta, but here you see, I just changed the scale in short, right? But the gamma has lower amplitude. From theta, I get the phases, right? So it goes from minus pi to pi. There is math behind this here, since this is a physics course, right? But I mean, we have to use Hilbert transform, and then we have to make this analytical representation of the signal. Anyway, there is a lot of math, but for those that are practical, we are talking about 1 to 2 lines of code as well in MATLAB, or whatever we want to program. And from gamma, we get the amplitude envelope, which is here. So the next step is to relate how this blue guy here, how this amplitude envelope it's related to the phase. So what we do is we beam the phase. I use 20-degree phases, so I have 18 phase beams. And to each phase beam, so say I have a phase beam that comes from this phase to this phase, I go here and go down here, and I see what's the amplitude for this phase beam. Whenever this phase beam shows up, here, I come here and collect the amplitude. In short, we come out with a plot like this, right? So this would be the mean amplitude of gamma as a function of theta phase. And I tend to plot 2 cycles just for clarity, because sometimes the amplitude is highest here, and then you can see anyway, but it's just 2 cycles. So notice that if gamma is not modulated by theta, I would expect to have a uniform distribution over here, right? Meaning irrespective of the phase, the amplitude of the gamma is the same. On the other hand, if there is some modulation, I would expect that something that goes away from the uniform distribution. And the modulation index I came out with is something very simple, and actually people use for other stuff besides this. But this would be a sort of Kullback-Leibler distance from the uniform distribution. Right? The MI stands for modulation index, if you guys want to look at the formula. But this guy is just computing how much this guy here deviates from the uniform distribution. And if it's equal to the uniform distribution is 0, I normalize it to assume values between 0 and 1. And in the perfect case, 1 never happens in biology, I should say, but the 1 would be achieved if the gamma would exist only in a single phase beam, so, and not in the others. This never happens, though. So the next slide is the most important. If you understand the next slide, you understand the rest of this part of the talk. So basically, what I did here, I got my original LFP. So I called raw signal, but it's an LFP signal, right? And I filtered in 2 pairs, in a pair, right? One frequency here and another frequency there. And out of this pair, I came out with a modulation index value. So this plot here is looking at several pairs, okay? So in short, okay, first explanation is whenever you see warm colors, this means that you are seeing phase amplitude coupling. More precisely, warm colors mean that the phase of the frequencies shown in the x-axis here are modulating the amplitude of the frequencies shown in the y-axis. So basically, say my 8 here denotes actually a signal filtered from, say, 6 to 10, but I plot the center frequency. And 140 here means someone, whatever, filtered from 130 to 150, but I plot the center frequency. I do all that framework, I come out with a phase amplitude distribution like this, I compute my MI, my modulation index, and I plot here as color. And blue colors mean that is the modulation index is low, so like plots like this give rise to the modulation index. So in short, warm colors would mean phase amplitude coupling between these frequencies here. So as simple as this was the tool, and then I start applying it. So just a review. Okay, so this is a collaboration with Anne Grabel's lab at MIT. So this is the first work I did there, my postdoc in this line of research. So basically, Anne Grabel, she's interested in the formation of habits, and she has a lot of recordings from the striatum. In this particular data set, we had striatum recordings and CA1, hippocampus recording. The task is a T-maze, Q-induced. So the animal goes through the maze, it has to learn to turn left or right. And when the animal starts listening to a tone, so this tone could be a high pitch or a low pitch, say if it's a high pitched tone, the animal has to go left. If it's a low pitched tone, the animal has to go right. Okay, so it's decision making based on the cue, based on a sound. And I'm going to show the results of this analysis. You're going to see these abbreviations here. Each abbreviation stands for the position of the maze where the animal was. Okay, you don't have to know by heart, but you just keep in mind that the animal is going through the maze. Going through the maze, pass through the gate, this is the start region, start to listen to the tone, starts the curve, finish the curve, and reach the goal. And we looked at these frequency bands for reasons that will be more clear, right? And in this work, I broke gamma. I mean, nowadays people are breaking gamma a lot, but by the time this was one of the first papers breaking gamma, not in a single entity, but in two. I mean, here I have low gamma and high gamma. Okay, so results. So far, I'm not showing you any cross-frequency coupling. So here, different columns means the power changes in different frequency bands. One row here is for the hippocampus, the other row is for the striatum. And within each frequency range, I'm showing you, these are already group results, how the power varied as the animal went through the maze, from the waiting there, from the warning until the goal. So the result here is that theta goes up and down, low gamma down and up, both high gamma and HFO, they tend to increase as the animals go through the maze. Okay? The same result was found to the striatum. And the punchline is we as neuroscientists, we don't break the frequency just because we want to break them. I mean, there are reasons for this. You see that theta behaves differently than low gamma. This is another reason why I broke low gamma from high gamma, because, I mean, the time course of power change, they're very different. Okay, so distinct frequency bands, they exist different variations as the animal goes through the maze. And now I'm going to apply that tool I showed you. So that was group result already, and this is just one animal. And okay, theta up and down, low gamma down and up, high gamma and HFO up as the animal goes through the maze. And then for each one of these periods, I computed that comodulogram. And the main result of this paper is this one here. Okay, focusing on the first row here, for instance. So what we have is the different maze, the different epochs of the maze. And remember, warm colors mean cross-frequency coupling. So the result of this paper is cross-frequency coupling appears and disappears as the animal goes through the maze. Okay. This one here, so something that changes with time. You know, we physicists we like to follow dynamics. So the title of the paper is Dynamical Cross-Frequency Coupling, blah blah. But it just is blue, blue, blue. Something are coupled, and then something is no longer coupled. This bottom row here is the same result as this guy here. Here goes up to 200. Here goes up to 100. It's just that there is a second island of modulation here, which is still statistical. I mean, this I did shuffling, and I removed all the non-statistical stuff. But anyway, but just to show that we found actually two islands of modulation. So the x-axis here is theta. Theta would modulate what I call as HFO, which stands for high-frequency oscillations, but would also modulate this guy here, which is from 80 to 100, which I call as high gamma. Okay, I'm going to tell you more about these two frequencies. But this is the main result of the paper. There is a dynamic cross-frequency coupling as the animal goes through the maze. This is the empirical result, and of course we like to think that this may have something to do with cognition and decision-making and stuff like that. But the main result, empirical result, is this one. A similar finding, we found this for the striatum. Actually, the pattern of coupling was different, and we only saw one island of modulation. Right? But it was also there, I mean, qualitatively, we had this dynamic coupling in striatum LFTs. Okay, this was the main result, and then we looked at where, so this is another analysis that we can do. So here, basically, I am filtering in different frequencies. I'm getting the amplitude envelope, and I'm computing the mean over each envelope, and this mean is triggered by, in this case, the trough of the theta. Right? In this way, we can see where in theta the oscillations are higher, and we normalize for each frequency, so we can correct for the 1 over f. So in this striatum, we saw that their island, they tend to be highest near the trough, right? You can see it here as well. So this one guy here, this 100, is a slice here. This is a slice, the dashed line is the reference theta, and we see that this 100 tends to be highest near the trough. Okay, it's shown in other ways as well. For the hippocampus, we got this picture here. So we saw the two islands, and one island tend to, so HFOs, they tend to be maximum at the peak, and the high gamma tend to be maximum in the rising phase. Right? So here is one slice at the 80 and 160, and we see that they have different preference. These are other analyses showing the same thing. One goes in the peak, the other goes in the rising phase. the reason why I like this result, I mean, the main result was that, okay, cross-frequency coupling, it shows up and disappears, and this would be a side product of that research. And in the end, so we have these different phases where these oscillations will show up, and if if you look carefully, this is an example, but there is statistics for this. The period in which this oscillation would be most modulated by theta deferred. So HFOs, they tend to be more modulated by theta during when the animal starts to listen to the tone, and high gamma, but when the animal finishes the curve. These we did some regression analysis, but in short, we computed how much the modulation of high gamma depended on the other frequencies. The thing is dependent on theta, dependent even on low gamma, but it was independent. The confidence interval cross zeros here of HFO. And vice versa, the amount of HFO modulation seems to be independent of high gamma. I'm talking to you about a side product of this research, but this is actually what I've been working these days, to try to characterize these oscillations here. But my point, I was all proud by this point here, because if you were, so this is a normal power spectrum, right? So here we have the different task epochs, task periods, the frequencies, and how things change, right? So we have the low gamma going down, and the high gamma and HFO going up. But my point being, if you only do this Fourier analysis, you can't separate out high gamma from HFO. I mean, they look like a continuum of frequencies. But if you start playing around with this, I mean, nowadays it's not that new anymore, but by the time this would be new tools for data analysis, you could identify that these things actually, they were distinct, oscillatory activity, right? So for this first paper, and I guess I will stop here because we are almost 4. Anyway, we found that we found the cross-frequency coupling. There was dynamics and everything like that. And then here we started to characterize this kind of activity, which are the HFOs. And by means of these new tools, you could clearly tell that they were not the same thing as gamma. And actually, you could also separate out low gamma from high gamma. And this continues, and then continues up to this day. But I will stop here because I'll let you guys rest. Thank you.
What is the biggest size of interaction matrix that you have worked with?
The interaction matrix will be as big as the number of cells that are involved in cell assemblies, right? And maybe it was for that human, because that I showed. I mean, it was just a preliminary result in human brain slice, and they had one of these things that they put in. I don't know. Do you know how they call? Michigan probe, maybe. The Utah probe. But anyway, I don't know how many recording points they had there, 50 or more. The Utah, yeah, 96, and then, I mean, it was huge there. I mean, I have to get back to the other one. A lot. but we are yet to do, say, a physiological discovery using these methods. I mean, so far we have these methods paper, but we have still to have the proper data set to see if we can figure out how the brain works with this, looking at Hebb's theory.
So you suppose it's only a matter of scaling up?
I'm sorry?
You think it's only a matter of scaling up, getting more neurons to do the
I
bigger
think
matrix?
it's a matter of getting maybe more, because this first data set, in the end, we realized that the animal was making some movements, like rhythmical movements, and it was just noise, but it can be rodents. It doesn't have, the reason why we didn't, well, maybe there is nothing there, right? But the reason why we spent 1.5 years and then we decided to drop all the analysis is because we thought that the activity of the units was contaminated by muscle, basically, by muscle activity. So we didn't trust, we're not that confident, no one wants to lie. So, but I think, I mean, if you can compute down to whatever, maybe you have more than 10 neurons, 10, 15 neurons, you don't need that many neurons. There are certain things, I mean, there are a lot of parametric stuff that I didn't show, right? I mean, if you want to your eigenvalue to be above this Marchenko-Pastur, I mean, there are some things that your cell assembly should have. So for instance, if your cell assembly spikes very little as an assembly, I mean, they may be more difficult. So if it's more active, either you record a long time to compensate for the fact that there is sparse firing, or there are things that you can do. It will also depend, actually it doesn't depend on the number of neurons, right? But depends on the spike frequency and how much they deviate from the baseline. I mean, how many times they get active and within each activation, how strong is the activation. So these technical details I didn't show you, but maybe you can have an example there you will not detect because it didn't look long enough or it was too weak to be detected.
Of your question, but we haven't done any paper with real data with this method, but lots of people have been using. We have distributed the paper for lots of different labs, and one person that was using was I can't remember his name right now, but he was in the Edelman Institute doing modeling, and he was applying these methods for this very large population of neurons that he had, but modeled, right? And he was not getting very robust results in this method, and we found out, like interacting by mail, that the problem was he had like 2,000 neurons, 3,000 neurons, and for this number of neurons, this method collapsed because that bimodal distribution he showed from the inner products was not bimodal anymore. So it was very hard to find the threshold to say, okay, this is 1, this is 0. But actually, this was one of the main reasons that triggered this new method that we developed later. That, as far as I know, he's using now, and
The ICA
it's much
base.
better.
It's PC plus ICA. Yeah, I'm trying to sell you an old car. We have a new one now. No. But it's nice to see where everything started and how we got here.
For 80%, let's say, of the data I've seen, they are exactly the same. But for like one case, like extreme case like this, and ICA, like this new method that's also based on ICA, is much more easy to understand why it works, and is not the computational time is not that large. So we are happy with it for now. We are like, there's at least 10 labs in the world right now working with our method. We just don't have the data to try ourselves.
Speaking of that, so does ICA give you overlap between neurons? Okay.
We have it here. Oh, anyway, you can talk later. Yeah, but it's actually it's ICA mixed. So this is the paper, right? It's ICA. It's mixed. We still need the principal components to. We still use the Marcinko-Pasteur stuff. Anyway, but it's a long story. I don't want to keep you guys. Maybe those interested can talk to Vitor and me. Anyway.
Okay, so maybe we can stop now, and if
Yeah.
there are more questions, you can follow up. So let's thank him again.
Okay, thank you.